Okay, so next we're going to talk about secondary structure of proteins. So we just finished talking about primary structure. So we said primary structure was the sequence of amino acids held together by peptide bonds. Also, something for you to pay attention to is that this is the only covalent interaction that you are going to see in these final protein structures. All of the other interactions between secondary, tertiary, and quaternary are going to be non-covalent interactions. So again, that's something you should always be thinking about when we're looking at how these molecules are being made, is what's a covalent interaction and what's a non-covalent interaction. So the primary structure is just the sequence of amino acids held together by the peptide bond. The secondary structure is determined by hydrogen bonding in the peptide backbone. So hydrogen bonding in the peptide backbone. So what I mean by that is these hydrogens that are attached to the backbone, this nitrogen and carbon chain that are available to hydrogen bond with each other. I'm not talking about hydrogens in the R groups. So these are all positions that hydrogen bonding can happen. Hydrogen bonding that would result in a secondary structure of a protein would only be these hydrogens that are attached to these amino nitrogens. And there's two major types of secondary structure that we're going to talk about. And there are some really good pictures in your textbook and in the PowerPoints. I'll draw some rough pictures on the videos, but you should definitely look at the textbook and the PowerPoint slides to see what they really look like. So there are two major types of structures. There's what's called the alpha helix. There's also something called the 3-10 helix. They look similar, it just is different, the rise and run of the number of residues per turn in the pitch and all of that. So when you look at the actual pictures in the textbook, you'll see that this alpha helix, there's going to be some hydrogen bonding between amino acids that are above and below each other in the helix, and that's what gives the helix this structure. So remember, the dashed lines will always represent a hydrogen bond or a non-covalent interaction. In this case, it's a hydrogen bond. And then the solid, this spiral, this helix right here, just indicates the sequence of amino acids. Another type of secondary structure, probably the second most common, is what's called a pleated sheet. And this looks like if you were to take a piece of paper and fold it up to make a fan. It looks kind of like that. And what happens is you will have multiple pleated sheets next to each other, and they will hydrogen bond like this, or they can hydrogen bond in what's called a parallel and an anti-parallel arrangement. So this way, where they're kind of right above and below each other, when this, so just to orient you, if this was going from, this is the N terminus, the nitrogen end, and this was the C terminus, and then over here this was N and this was C, we call this an anti-parallel beta pleated sheet. Or you can have the opposite, where you have bonding, but it's a parallel, meaning that these are both going in the same direction. Parallel pleated sheet. So the shape that this secondary structure of the protein adopts has to do with what's called phi and psi angles. So the phi and psi angles are on either side of the alpha carbon. So one thing that's important to remember is that the peptide bond, let's just draw the peptide bond again. So the peptide bond, that is, so this would be the peptide bond right here. This peptide bond is rigid, and again, I'm just going to do glycines. It's almost planar. It actually is in a resonance structure. And there's no rotation around this bond, because this is actually a resonance structure where this bond acts like a double bond. This peptide bond acts like a double bond, which means it is rigid. So there can't be any rotation here where the peptide bond is. Where there is rotation is on either sides of the alpha carbon. So here's the alpha carbon here and here. So what we do have is we have rotation around between this nitrogen and this carbon and this carbon and this carbon. We have rotation around these bonds, but no rotation here where the peptide bond is. So on the nitrogen side of the alpha carbon, that is called, so right here, that is called the phi bond. And then on the carbonyl side of the alpha carbon, this is called the psi bond. So again, here we would have phi, and here we would have psi. So depending on the angle of rotation on these phi and psi bonds on either side of the alpha carbon, that will tell us what type of secondary structure a protein will have. And we're able to plot these angles of secondary structure to predict what the secondary structure is going to be, if it's going to be an alpha helix or a pleated sheet, anti-parallel pleated sheet, using something called a Ramachandran plot. You don't need to know how to use these or how to read them. You just need to know that what a Ramachandran plot is that it's a plot, it's a map of all of the phi and psi angles in the region of a protein. And based on the location of the signal on this map or this plot, you would be able to predict what type of secondary structure a protein would have. So for example, up here, if you have a lot of angles that are within this region, then you would be most likely to have a beta strand or beta sheet, strand sheet, same thing, anti-parallel or parallel. If you were in a region down here where you had phi and psi angles that were in this region, you would be more likely to have a helix, right-handed helix or a 310 helix. Over here, these are left-handed helix. This would be a left-handed helice. You are not responsible for knowing the areas of the plot that indicate different types of secondary structure. What I want you to know is that a Ramachandran plot is something that shows what type of secondary structure is predicted based on the phi and psi angles in that protein structure. So the protein, once it achieves a secondary structure, this rotation will then predict what type of secondary structure the protein will have. It shows, and that plot predicts what is most energetically favorable. So again, this is at the beginning of chapter 6, both in your textbook and in the lecture PowerPoints, and it goes through this in a lot more detail. You're not responsible for memorizing any of the specifics. I would want you to be able to tell me the two major types of secondary structure, what a Ramachandran plot is, what are phi and psi angles, where are they located in the protein, that kind of stuff. So secondary structure is based on hydrogen bonding within the backbone. So nothing to do with the hydrogen bonding that is on any of the R groups. We have not talked about that yet. Okay.